And again, this was a huge revolution. So through the 70s to the 2010s, probably, it was sort of the dominant paradigm of cancer. It's a genetic disease. And what happens, of course, is that now you can develop new treatments that attack this new paradigm. So instead of developing drugs that are just indiscriminate killers of cells, you can now design drugs that correct the genes that cause too much growth. And the first few drugs that came out, so there's one called imatinib, there's another one called trastuzumab for breast cancer, were just like revolutionary. Like the late 90s, people were like, this is incredible because you could give these drugs without a lot of side effects because, these drugs were changing these genetics. They weren't indiscriminate killers of cells. So then what happened, of course, is that with these first couple of drugs, everybody's like, wow, we're going to cure cancer. This was the sort of prevailing thought by the 2000s. All we need to do is figure out the 2 or 3 mutations that are critical for these different types of cancers, develop the couple of drugs to fix them, and boom, you've cured cancer. So you're going to develop one for breast cancer, you're going to develop one for cervical cancer, you're going to do this and that. So the problem was that, and this was called the 2-hit hypothesis, that is that you had a couple of genes that were critical and you hit 2 of them. And the reason that smoke, for example, tobacco smoke was causing cancer was not that it's a targeted gene damage, but it was that if you have a lot of damage, by chance, you'll more likely get those 2 that hit. But the problem was that we looked for these genes. So we did the Human Genome Project. In the 2000s, we mapped the genome of an entire human being. And then we weren't much closer. So then we did the Cancer Genome Atlas, which was an even more ambitious thing. Instead of looking at the genes of one human, we're going to look at cancer cells. And we had like 33,000 different genomes. And you just compare them and figure out which ones are good or bad. So the problem was that at last count in 2018, we identified about 6 million different gene mutations in cancers. So cancer didn't have 1 or 2 gene mutations. It had like 100. So if you had, a cancer clinic, so somebody with colon cancer, patient A with colon cancer, he had 100 different mutations, and patient B sitting next to him with colon cancer had 100 mutations that were actually completely different from his neighbor. So this, of course, made it impossible to go any further with this genetic sort of paradigm, because you can't develop 100 drugs for patient A and a different 100 drugs for patient B, even assuming you could afford it and they had no toxic side effects. So that brought progress in cancer medicine basically to a screeching halt. So this paradigm that we had used to look at cancer, which we were developing drugs, these genetic targeted drugs, really didn't work. Like the number of drugs that have been developed and useful are like, you count on one hand probably. And this is after 30 years of good research. Like we spent a lot of money on research.